and team for the invitation to speak today. We're really delighted to tell you a bit about the genetic research in PCD. And I am joined today by Motile, who has been doing her PhD here in London for the past year. So we're going to tell you about genetics in PCD diagnosis, and particularly two genetic initiatives, the CiliaVar database, which is part of Work Package 2, and the ClinGen collaboration that the BEAT-PCD Work Package 2 team have set up. So just a one-slide intro on who we are. So Motile and I are working at University College in London. We work at the UCL Great Ormond Street Institute of Child Health. So we're next door to Great Ormond Street Children's Hospital. This is our team, and some of you may be familiar with the other PCD fans in our building, Steve Hart and Chris O'Callaghan, who come to meetings, and there's Motile and I. So together with Great Ormond Street UCL, ICH is one of the largest concentrations for translational health research for children. And I wanted to credit here up front the BEAT-PCD Work Package 2 team. So Work Package 2 is about the network of research databases and collaborations. And here we are. And important collaborators are the University of Dundee Health Informatics Centre, who do the IT for the CiliaVar database. So I know that I really don't have to introduce PCD, but here's the image that we're very familiar with from Toronto Sick Kids to show the problem in PCD, which is a problem with the cilia not moving properly in our airways that stretch from our nose right down to the gas exchange areas. We now understand that the incidence of PCD, which is a congenital inherited disorder that we see in families, is at least 1 in 7,500, and that's very likely still an underdiagnosis. And the essential problem is that the cilia don't move, and in the airways that means decreased mucociliary clearance, leading to recurrent airway infections, lots of inflammation and nasty bacterial colonization, and a sort of inevitable path down to the loss of lung function, bronchiectasis. The location of other cilia means that PCD patients can have laterality defects and infertility as part of the phenotype as well, which is important for genotype-phenotype correlations. So it's clinically heterogeneous. It's also genetically heterogeneous, as I'll talk to you about in these few intro slides, with at least 60 genes associated to PCD so far. So PCD is a monogenic disease. People often wonder whether a variant in one gene and a variant in another gene can combine to create PCD, but we have no proof of that. So PCD is a single gene disorder. So variants in any one of those 60 genes can cause disease. It's primarily a recessive disease, so where the two carrier parents have an affected child with contribution from the maternal and paternal chromosomes. And in the UK, we think, certainly in London, there's probably about 40% recessive consanguineous families. So this is where there's a common ancestor between mother and father, often first cousin marriage. So those are the recessive forms of PCD, but for a few genes, we now recognize that there's X-linked PCD and there's autosomal dominant PCD. So in both of these cases, it's a different situation, and it's also heterozygous variant, so a single copy in an X-linked gene or in a dominantly acting gene can cause PCD, whereas the recessive forms all have biallelic variants. So just a note to say that the motile cilia in humans are on incredibly specialized areas of the body, on the epithelial surfaces or moving the gametes around. So we have the multicilia of the airways, but we also have multicilia in the fallopian tubes, in the brain ventricles. We have the monocilia that are very important during embryonic development, and cilia are highly connected to sperm tails or flagella. So that means, as well as this is the kind of a very crude summary in a review to show that in PCD patients you get upper and lower airway disease, but in certain cases there can be hydrocephalus due to the brain cilia not working, and then subfertility with the sperm and the eggs not being moved correctly. And laterality is randomized in patients, so 50% of people are going to have laterality defects with their internal organs being placed in the incorrect wrong side. And in a proportion of those, that leads to congenital heart defects. And when you have a heart defect, very likely your PCD disease is going to be worse. So the problem is the motile cilia. You take a nasal brushing and you can make quite a nice model of what's going on in the lungs and in the airways of PCD patients. And what you see is this sort of picture that I know that most people in this audience are very familiar with. So in a normal, healthy human, your respiratory cilia here, you see the beautiful metachromic wave of the cilia. They beat 1 million times a day, and you can see the mucus flow on top. Around 12 hertz. In PCD, what you see there is static cilia. So the video is kind of shaking, but the cilia don't move. And what we're looking at here are the ciliated cells. So ciliated cells are sitting there in the airway epithelium alongside the secretory cells that produce the mucus, with the basal cells at the bottom, the stem cells. Oops. So, cool. Hi. Somebody speaking? Or maybe that's me. Okay, so if you look inside a cilium, it's quite a complex structure. So poking out of the top, we get the microtubules that stretch all the way from the base to the tip. And the microtubules have proteins attached to them. And if you cut through a cilium, this is the 9+2 array of cilia. That's this highly conserved structure. And these are the structures that we are familiar with, because these are what goes wrong in PCD. The dining arms, outer and inner, the radial spokes, the central pairs. These are the microtubules that have other proteins attached, particularly on the inside. We now know there's inner microtubular proteins, or MIPs, the necks and links. And in a healthy person, this is what you see when you do that cross-section in conventional electron microscopy. And there's a PCD patient. And what you can see in this case, it's probably one of the most common defects in PCD, is loss of these dining arm motors. And those are the things that make the cilia beat. So when they're gone, the cilia do not beat. This is a slide just to say that it's more complicated than that. So if we look at this, all of these peripheral microtubules, the 9, we're going to see that the outer dining arms and the inner dining arms and the radial spokes and the MIPs, these are all multi-subunit complexes. So there's a lot of proteins in there, and you can imagine those going round, making quite a complicated picture. And then these motors are distributed at 96 nanometer repeats all the way along the cilium. So in the 96 nanometer repeat, that's what you see when you do this fantastic cryo-electron tomography. So my point here is that these are complicated organelles. So the genetics is complicated. So in proteomic and genomic studies that have been done, there's been done a few of the human motile cilia and many in model organisms. So there's maybe 700 to 1,000 proteins that make up the cilium. If we say loss of function variants causing PCD affect about 60 of those, we can well imagine that 60 may be a low estimate, and we're going to be continuing to find PCD disease genes. It still remains the case that all the genes that are mutated in PCD are involved in the growth of the cilia, in transport of proteins to the cilia, and probably their entry into the cilia. They're moving along the cilium to be distributed along the axoneme, and these structural defects in these structures that I've shown in the dining motors and the radial spokes. So just briefly to say that there's, although we have a complex picture, one of the great things about PCD is this remarkable correlation that we get between a specific EM defects that you can see. So there's outer arms missing, inner and outer, or inner arms gone, radial spokes gone, transposition defect where the central pair is gone and you get transposition of a peripheral microtubule. These all are correlated to predictable changes in the motility. And then what we can also see is starting to emerge some really very strong correlations to the clinical course of PCD patients. So we know that a couple of genes that are involved in that ruler, in that 96 nanometer repeat distribution, those cause a very severe form of PCD. There are certain genes that affect, for example, the central pair and the radial spoke. Those you won't get laterality defects because the developmental node cilia are not affected in these cases. So this isn't a lecture about all those different correlations, but really the point is that the underlying genetics connects to the structure and the functional defects, and also starts to become predictive for the phenotypes and the long-term outcomes for these patients. For example, if you're not going to get laterality defects, you are not going to get a congenital heart defect connected to your PCD, that kind of thing. And we know that some of these genes are not involved in fertility and they're not needed for movement of the sperm. So these kinds of correlations. So that makes some really fantastic diagnostic tests for PCD, but none of them are 100% gold standard. So when a PCD patient comes into clinic, they have low nitric oxide in their noses. You can take the brushing and you can do genetics. From nasal brushings, we can look at cilia beat and waveform through high-speed video imaging, and then electron microscopy, and with the supporting immunofluorescence staining of ciliary proteins, we can look for these structural defects. And so there are a couple of ways that a single test can be diagnostic on its own for PCD and give you a diagnosis and a report. So class 1 EM defects are 3 at the moment: loss of outer dining arms, loss of outer and inner dining arms, and microtubular or transposition defect where the microtubules are affected and the inner dining arms are gone. Those are diagnostic. From genetics, if you have a biallelic, so one from mum and one from dad, biallelic variant inherited in a patient in a recessive gene, or if you have a single variant in an X-linked gene, because you just need one copy to be affected in a male, so that's just a few genes. And then we have these dominant forms where again you just need one copy. FOXJ1 and TUB4B are examples. That's also a diagnosis. Where we hit issues and where the things that we're going to talk to you about in this talk come in is really where there's inconclusive test data and we need more evidence. So there are class 2 EM defects, central pair loss, ciliogenesis defects, where you have microtubular disarray, but you retain your inner dining arms, or if you have partial losses of your dining arms, your outers or your outer and inner. These are all not diagnostic on their own, and they need to have concordant genetic data or cilia beat or immunofluorescence data to be diagnostic. And about 20% of cases, you just don't see a defect that you can register on conventional TEM. Those are also inconclusive. And then on the genetic side, we have these things called variants of uncertain significance, or VUSs or VUSs. So what is a VUS? So Mafalda in the second half of this talk is going to tell you more about VUSs. If I just say that at the moment a VUS is a variant that you've detected in the DNA of a patient through patient sequencing, exome sequencing, or gene panel sequencing. but you don't know if it causes disease or not. It hasn't been reported before, for example, or the pathogenicity of it is unclear. So a couple of slides on the VUS issue and why this is a problem in PCD. Essentially in PCD, there's many uncharacterized, extremely rare, often family unique variants that have been found through PCD patient sequencing. And diagnostic centres and clinicians and researchers then struggle to understand whether that variant is really causing the disease or not. And to illustrate this, I've just taken a snapshot from a tool that people refer to. This is really one of the most commonly used tools. It's a public repository of clinical variants. So this is called ClinVar, and I'm sure many people in this audience access ClinVar, if not on a daily, but on a regular basis. So anyone can submit their variants to ClinVar. So the ClinVar team curate variants taken from publications, PCD genetic specialist centres and commercial gene test labs all submit their data into ClinVar. So they sequence a patient that they think is PCD or is confirmed PCD by associated tests. They get a gene test result and they put it into ClinVar. So sharing of these genetic data is absolutely vital for everyone to get more knowledgeable about what is or isn't a variant that causes disease. But just to show the issue of PCD, so this is a snapshot from yesterday. So there are multiple variants that have been found and reported into ClinVar, deposited into ClinVar from patients, 30,000 plus. The classifications are variable. So Mafalda again will go a bit more into how we classify variants. But so, for example, there's 2,400 that are classified as benign, 13,500 likely benign. So these really aren't supposed to be disease causing. There's 9,000 that are uncertain, and then pathogenic and likely pathogenic, there's much fewer, as you can see. I mean, it's good to have nearly 5,000, but there's less with this very clear-cut pathogenic prediction. And there's various different types of variant and larger variant types. And you can start to see that variants may have multiple submitters, and that might turn them into something that's more judgeable to be pathogenic. The more people that report it connected to PCD, it sort of adds to the evidence. But 22,000 times there's just one submitter of one variant. And there's all these conflicting classifications. So ClinVar is a completely key resource. It's absolutely important to have this kind of thing for genetics interpretation, but it's not highly evidence curated by experts and experts in PCD. And the other point I wanted to make comes from this terrific paper that was published in 2022. It's a massive big data paper from the Hannah et al. Group. And what they did was they took disease out of the equation. They weren't looking at PCD patients. In fact, they removed PCD patients. And what they did was classify variants in known PCD genes simply using variant classification guidelines that Mafalda will expand upon. And so this is the paper that tells us that overall the prevalence for PCD is 1 in 7,500. That's why we say that these days. But my point here is that that's quite a cautious estimate. So when they looked in the NOMAD database, which is a big database of non-PCD cases, and they look for kind of hidden variants and disease-causing variants to look at the incidences, and then they compared it to genetic test results from a PCD panels, then they could really get a good estimate. But this is their estimate excluding VUSs. So if they disregarded any variant that was of uncertain significance, so wasn't a likely pathogenic or pathogenic predicted allele, they got this incidence. Then they did the same thing again, but they included all the variants of uncertain significance in a set of known PCD genes. And then suddenly the variance went up enormously. Overall, you could even claim, based on using VUS data, that 1 in 127 people have PCD overall. And that's really just to show how what a big problem VUSs are. So very many of these are actually going to be benign or predicted benign or likely benign variants. But they're in PCD genes, so we have to kind of weed out the VUSs. Okay, so there are challenges in PCD genetic diagnosis. We have multiple genes, over 60 so far, and we don't have all the genes. Gene testing is not a gold standard, so we might end up with 100 PCD genes. They do keep emerging in the literature. So gene panels really continuously need updating to be up to date and not risk not screening of new genes or having genes that are not really linked to disease on them. So a lot of updating. As mentioned, there's very many variants. It's been estimated that 60% of variants found in PCD patients are actually just unique to an individual family. They're so rare, and that means that the supporting evidence is just from single families. So you have to look at very many families and put them together and find variants in a single gene to make conclusions. Then based on CiliaVar, which is a database that Motile was going to tell you about in the second half of the talk, we estimate that 20 to 30% in variants found in sequencing of really very well characterized PCD cases are VUSs. So we're not completely sure that they're disease causing. I just mentioned, but I haven't told you anything about it, that there's a particular large gene in PCD. It's one of the central pair components. It's called hydin, and when you have a loss of function variant in hydin, you're going to get PCD, but there's an almost identical copy gene in a completely different place in the genome, different chromosome, but it overlaps massively with hydin. So calling variants out and knowing that they're unique to hydin and they're not in hydin 2, which is not connected to PCD, that's a very specific problem in PCD. So, and then generally, our genetic challenges are the complexity of the cilia biology that I've told you about, and really our kind of lack of full genotype phenotype understanding. So when we get a variant in a particular gene, it's not you know, the phenotypic connections are still need a much better understanding to be able to do the genetic interpretation. So that's a lot of problems. So having a positive test, to have a genetic test that really diagnoses someone with PCD having biallelic variants in a very well understood gene and they look pathogenic, this is really wonderful and gives some really unique advantages. So you have a molecular diagnosis. You can do family planning and you can do carrier testing. And you can't do that with other diagnostic tests for PCD. You have to have your genetic results to be able to do these kinds of things. As I sort of briefly explained, your genetics can predict some aspects of your disease course if you're a PCD patient, things like severity, and can hopefully increasingly direct the clinicians taking care of patients to the best types of management and the most appropriate types of disease management. Your genetics can predict the involvement of organs other than the airway, so you can predict things like fertility and hydrocephalus and laterality problems. But one of the really main important things, particularly for people in the genetics field, is the absolute excitement to see nucleic acid therapy trials are now proceeding with companies like Recode. And of course, it's an essential requirement if you're a PCD patient to know your genetic result. So Recode are focusing on DNA-I1 gene as their first product, and Ethris are focusing on CCDC40. Those are two examples where to get into that trial, you would have to know your genotype clearly. So it's super important, and I'd say increasingly important, that we get genetic diagnosis correct and we start to resolve some of these so-called VUSs. And that requires a systematic approach to look at causality of the PCD genes. Is there really good evidence that a problem in a particular gene will give you PCD? And then also to do the same thing, looking at the causality of the actual DNA variants that are found in genes in genetic testing. So we are going to talk to you today about two really exciting initiatives to improve PCD genetic diagnosis, the ClinGen project and the CiliaVar project. So ClinGen is really about first needing to establish what are the causative genes for PCD, and I'm going to tell you about ClinGen. Then I'll pass over to Motile, who's going to tell you about CiliaVar, and this is our brilliant BEAT-PCD project. So this is a tool to allow us to have a better idea of what variants in PCD genes actually cause or don't cause disease. So ClinGen, or the Clinical Genome Resource. This is a fantastic initiative. It's a USA NIH-funded initiative. It's been going for many years, and it's aimed at defining the clinical relevance of genes and genetic variants for use in precision medicine research. So it's really addressing the problems that I've mentioned, and many diseases are being looked at, and there's many groups in all sorts of other conditions who are doing the same as us for PCD. But so this is a gene disease validity curation. So we're looking at the genes at the moment, and we have set up the Motile Ciliopathy GCEP. So that's the ClinGen word for a gene curation expert panel. It's been going actually for 4 years, established in 2021, with 28 active members, and there's 10 biocurators. So we have Christina is our coordinator, we have the leadership team. The curators are massively important and hugely hardworking individuals. They're all trained up in how to look at the genetics and look at the connection to disease. And then we have our other 17 members who are the experts, including the leadership team. So these are clinicians and researchers who work in PCD and can really input vitally important information, because many of the biocurators are really quite new. I mean, they're not so new, they've been doing it for 4 years, but are new to PCD when they join the group. So they undergo a specific ClinGen highly specialised training. So we've been meeting every month, and we've been curating approximately 70 genes for PCD. And as part of this work, we've also curated genes for sperm failure or sperm defects, hydrocephalus and heterotaxy. So ClinGen is all open access, and I encourage you to go and take a look. So the ClinGen gene curation process is that the gene list, these 70 genes that we've selected, those are selected by the group, by the experts. The biocurators for every gene undertake a really detailed review of the genetic literature that references that gene. And then they do evidence scoring. How good is the evidence that that gene, when it's defective, when there's a loss of function variant in it, is really going to cause the phenotype of PCD? And then we classify variants according to evidence scoring, and there's 6 possible scores: definitive and strong. Those are the really strongly connected genes. Then we have moderate and limited. and we have fewer where we can actually find no disease association, or it's disputed or refuted. So at our meetings, the experts and the whole group review the curations done by the biocurators, and we reach a consensus. And that means this is why we've been going for 4 years, because a whole meeting can just be devoted to one gene. It's a really very detailed process. And then once we're all agreed, we take a vote, and we have a public database publication on the ClinGen website of our curation results. So there's two sort of essential types of evidence: the genetic evidence and the experimental evidence. So when the biocurators are scoring genetic evidence for the link between a particular gene and PCD, they're looking at case studies and segregation data, whether parental genotypes have also been obtained, and they're looking at functional impact of variants. So it's kind of in silico functional impact, whether it's going to cause a frame shift or a missense, that kind of thing. Then in PCD, you can get a real range of experimental laboratory evidence, or I guess in silico evidence as well. For example, and this is ploughing through the literature to see whether the biochemical function is understood of the candidate gene or protein, whether it interacts with known disease-related genes or other genes, and it makes sense as a Motile ciliary component. Then variable data on animal and cellular models. Many different models are used in PCD research: Chlamydomonas, mice, zebrafish, and quite if you're lucky, there'll be some sort of rescue experiment. So restoring function in knockout models has been done. So all that evidence has gone through with a fine-tooth comb, and I hope people can see this. It's just an example that I've thrown up of GNH5. So GNH5 is one of the very original PCD genes to be discovered way back, and it's one of the most commonly mutated, although it's one of the bigger genes, and it's one where overall it causes the most proportion of PCD variants in DNH5. So this is the scoring that's been done by the biocuration team, split, as you can see, into genetic evidence and experimental evidence. And again, this is all freely available. So for genetic evidence, as mentioned, you look at all the family data that's been published, the genotyping in human families, and how convincing that is, and there's a possible maximum score for the types of variants and whether the segregation is fitting, and so on. So DNH5, unsurprisingly, because there's very many variants and very many excellent reports about DNH5 by now, that scores a maximum of 12. For the functional evidence, these are maximum scores for biochemical, protein, and expression data, then functional alterations in actual human patient cells, and then looking at disease models, non-human model organisms or cell culture human models, and then looking for rescue. So of a possible 6, this DNH5 has scored 6. So that's not surprising, and in a way, or in this case, the bio-curator would have had, I'm sure, the possibility to score it much higher, but those are the maximum scores. So what gets published is a definitive connection between PCD and DNH5, and there's a beautiful report summarising the evidence that's put out onto the ClinGen website. So there are less well evidenced genes. These are the classifications. So I've just listed as examples some examples of disputed and limited published curations of genes when looking at whether they cause PCD or not. So limited and disputed is really quite low evidence. So why have we scored these as disputed and limited? So I just take one example here, DNH8. So DNH8, just like DNH5, is a really big gene. So it has a possibility to have lots of variants in it. So we curated, the group curated DNH8 because there is a report that links it to PCD. There's actually only one paper so far, I think that's still the case, to say that DNH variants are a possible cause for PCD, but that's very limited data. And what the other piece of information about DNH8 is that it's not really expressed in the airways, but it's highly expressed in sperm. And DNH8 is actually much more highly linked in the literature to morphological abnormalities of male sperm. So it's much more linked to male infertility. So then the group makes something called a lump or split decision. They go through the evidence for both of those phenotypes and decide, is it possible that variants can cause both PCD and sperm defects, or should this be split? And we just consider DNH8 as something that causes a problem with your sperm and not your airway. So the decision was made to split. And then what you can see published on the website, and you can go and see the detailed report, is that it's disputed. So it's really bottom rank. It's really, you know, we couldn't say there's no disease association because it has been reported, but that's disputed. So that's a very, very low score. Yeah. And then when you curate for sperm failure, so-called sperm failure for DNH8, it's strong. So it's actually got a really top score. So you can start to see systematically going through genes to see whether they're connected to PCD. Okay, so to summarize where we are with the GCEP, the GCEP has published 63 curations for 56 genes so far. And I think given that that's a 4-year period, I think you can start to see what a kind of detailed process it is with huge amounts of discussion, people talking about their own cases and all sorts of things come into the mix. So we have less than 10 genes left to curate, and we're hoping to have a manuscript submitted to describe the GCEP work for this year. And we can make conclusions already about which genes that we would like to include or exclude from PCD testing panels. We would strongly say that DNH8, for example, should not be on a test panel for PCD. You can put it on a male infertility panel, and if you have that knowledge, of course, but not for an airway disease PCD panel. And there will be recuration. So we haven't actually started the recuration process, but as new evidence arises, new data on genes, a gene can turn from being disputed or low evidence into having better evidence as new publications and information arises. So it's important to say that the curation is not final for sure. And we're also working with ClinGen on variant curation. So the VCEP is something that's a kind of expansion of this work. And I've just got one slide that I think I have 2 slides left. So just talk to you about the VCEP. So using the same principle, the VCEP is a curation system that's supported through ClinGen. And this was established a bit later in January 2023. So it's been going for a couple of years with Marie and Maimuna as the chairs, the same coordinator who works extremely hard with us. Christina does an absolutely terrific job for the GCEP and VCEP groups. So this is now doing the same job, but looking at a much bigger job, which is curating all those hundreds and hundreds of gene variants. So how are you going to tackle that? And sorry, just to acknowledge again the list of curators and the experts as well, many of whom will be known to this group. So the VCEP team have kicked off by adapting the variant classification guidelines. So Mafalda will go into the ACMG guidelines in more detail. I'm just here to say that they've developed a PCD specific variant classification rules with all the expertise that we know about PCD. If you get a variant in this gene, you're expecting this particular structural defect, you're expecting this particular motility defect. So all those really nice correlative things help us to classify PCD variants. So they picked DNH5, the most commonly mutated gene, as their first example. They've developed the rules already, which is an absolutely terrific progress, and that's been submitted to an independent ClinGen committee, the Sequence Variant Interpretation Committee, who fed back what they thought about the PCD rules, and I think there's been 2 feedback sessions where these have been addressed. So we're really feeling ready to go with that. 50 DNH5 variants selected for a classification pilot run using these rules. And the advice really was to go to focus on variants, not variants of really clear-cut causes, but to look at VUSs in DNH5 or those with conflicting pathogenicity interpretation. One person thinks they're likely benign, person thinks they're likely pathogenic. So to really delve down into these rather tricky variants. And the group have conducted several sort of very educational mock curations already. The pilot curation is planned to start this year, once the classification rules have been pinned down by the SVI working group. So in my final slide, it's just really to point out that the BEAT-PCD works in a really important network of international collaborative projects. I've just listed a few here. I'm sure I have not listed your favorite one, so apologies. I was just trying to think of initiatives that I could think that really focus on genetics. Should be ClinGen as well. And of course, working closely with the other networks, the clinical centres and hospitals taking care of the patients and the families themselves, so their support, the support charity advocacy groups, other rare disease networks, and pharma partners. So, for example, Recode have been funding patient sequencing. So it's a really kind of big network, and these are essential to pull together this complicated PCD genetics information from around the world. So that's my final slide, and I'm going to hand over to Mafalda.
Thank you, Anna. Just going to share the slides. Can people see my slides?
Yes, we see them.
Yes.
Perfect. So, thank you. Just before I talk about the CiliaVar, which is the variant database, which is the main topic that I will talk about, I just wanted to share some data that really shows the importance of the GSEP and what Hannah's been talking about how important it is to get to curate the PCD genes and make sure we know which genes cause PCD versus which ones do not. So we sent out a survey via BEAT-PCD to a number of PCD centers across different countries, and we asked the questions about how they perform genetic testing. And what we learned is that actually many PCD centers use gene panels. So you can imagine how important it is to really know what genes to include in those panels. So 11 centers, actually, there was about 60 centers that responded, but 11 of them actually provided their own custom gene panels that they create and they use in their centers. But other centers actually use commercial panels, which are available by companies. So we did do some analysis to check both from the commercial panels, but also the custom panels, what genes they include that we have validated with the GSEP. So this here shows 33 commercial gene panels, and at the bottom we have the genes that we have curated with the GSEP, and they're color-coded by the classifications. So the ones in red, pink, and orange are the ones that we would want to encourage to be in PCD panels, but the ones in the 2 shades of blue, we really wouldn't. And as you can see, there is a lot of missing. So the bar charts go up to 33, so that would mean DNH5 less than 33 panels include the gene. So there's a lot of genes that are actually missing from all of these panels. And interesting enough, TP73 is actually a gene that we have recently validated as strong, and none of them included. But I do want to say that this does include some panels that are not specific to PCD as well. So when you do look at PCD specific panels, and there were 17 of them, so these are all here on the left, and they are from labs that are quite well used, such as Invitae, Mayo Clinic, GeneDx, and Blueprint Genomics. The distribution does look a bit better. We have a lot more inclusion of the red genes, which are definitive, as the previous slide, but we do still are missing a lot of these genes here. For example, you can see these pink and orange and red genes here. They're really not included in many panels at all. And just lastly, these are from the custom panel. So this distribution looks way better because you can see a lot more blues that are not being picked up, but a lot more reds that are being picked up, but it's still not, they're still not perfect. For example, TP73 is really not included in most panels as well. I guess this just goes to show how important it is to classify these genes and then for panels to be encouraged to include them and update depending on what the GSEP publishes. So before I go into talk about the variant database, I'm sorry because I'm sure most people here do know what a variant is, but just in case someone doesn't, a variant is a change in DNA sequence that is compared to the standard reference genome. So the reference genome doesn't come from one specific individual. It's an aggregation of many individuals, and you come up with a baseline. And then whatever variant an individual might have that is different from that baseline is called a variant. So variants don't always mean disease causing. Most of them are actually harmless. Some can cause disease, and some, as Hannah was saying, is the VUS problem. They have unknown effects. But based on a variant itself and the position on the protein, you can already know the consequence in that protein for some of them. So for example, here you have a wild type, so this would be the reference. And if you have a G, that gets translated to a serine in that amino acid position in the protein. But if you have a variant and instead of a G, you have an A, this can change that amino acid in that position. This can be the case for some variants. Other variants, you can still have an A and have the same amino acid protein, so that would have less of a detrimental effect on the protein. But on the other scale, you can have the more severe variants where this change would here mean that you would have a premature truncation of the protein, which means the protein that gets produced is shorter, and in some cases can be non-functional and cause disease. So there's 5 classes of DNA variant classification. So you have variants that you can classify as pathogenic or likely pathogenic, which are class 1 and 2. And these would be clinically significant. So if you have a pathogenic variant in a PCD gene, that could be diagnostic. You have class 3, which are the tricky ones, variants of uncertain significance. These are the ones that we want to tackle, and our database really wants to tackle these ones that I will talk about. And then you have class 4 and 5, which are benign and likely benign, which means these variants are harmless. So we don't really want to look at these variants in our database. So how do you get to those classifications? There's ACMG guidelines. These were actually published, I think, 10 years ago now. There has been some updates to it, but this table is from 10 years. So there's things have changed a little bit. But ACMG is the American College of Medical Genetics and Genomics. And they came up with this matrix where you can apply different rules based on the evidence that you have for a variant. So on the left, you have different types of categories of evidence. There can be population data. There can be computational and predictive data. So these are tools online that can predict an effect of a variant on the protein. Functional data like in vitro studies and so on. And you can have different types of evidence codes that you can apply, either that support the variant being benign or that support the variant being pathogenic. So one example of a very used evidence code is the one on the right, which is PVS1. And this is what I was talking about when you have sometimes a variant that you know causes a premature truncation of the protein, so the protein is shorter. And in some genes where you know that loss of function is a mechanism of disease, you can always apply this rule, which is the case for all PCD genes, I believe. So for example, if you apply this rule, and another rule that is very, very common, especially in PCD since variants are so rare, is this PM2 rule. And this one has a little bit less of strength. So this one's moderate strength. And this one you can apply if you have a variant that is absent from gnomAD, which is it's the genome aggregation database. So that's a database where you have, I think, hundreds of thousands, if not more, genomes from healthy individuals. So these are individuals that are selected to not have rare disease. So if you have a variant that is absent there, it means it's absent from general population. So usually rare disease, rare variant. So if you apply, for example, these two rules, that would already make your variant likely pathogenic, and that would be diagnostic. So there is, I've put the link, but there is guidelines to how you can apply these different rules. It gets more complicated than this. And these are the rules that the VSEP are trying to adapt to be PCD specific as well. And once you apply different rules, you can kind of score them and then come up with the final classification for your variant. And another thing to bear in mind is that if you apply, for example, some rules for benign, but some rules for pathogenic, this is conflicting. So usually these variants will end up being a VUS, a variant of uncertain significance. So now I will talk about CiliaVar, which is our variant database for PCD. This will be the first open access database of PCD variants. This is international. And the main aim is to improve the diagnostic accuracy of PCD. So to have all of the variants that have been published and also in some clinics and be in one place. So we do include VUSs because the more we look at them, the more we can be able to maybe reclassify these VUSs as pathogenic or not. So we currently have almost 2,500 patients, and all of them have our genotype. So we know what gene and what variants have been associated in this case to be causing PCD. We have a large number of different variants. And we also have clinical and diagnostic data that link to the variants for each patient. So some examples of clinical data that we include is upper airway symptoms or lower airway symptoms like bronchiectasis, fertility status, and so on. And we also, the diagnostic data will be things like if there's structural defects in the cilia or motility defects in these patients' cilia. So where did we get these patients? So most of them are from the literature. So we did a PubMed search where we searched for the gene and we tried to find all of the case reports of PCD patients with variants in that gene. So we do this on a gene by gene basis. But we also, about a third of the patients actually come from diagnostic centers. So far, 6 centers have contributed with data because they had the ethics to share and they wanted to. And usually when we get patients from the centers, we do have richer data in terms of clinical details. So as you can imagine, when we get data from the literature, you can't always trust what's published. So a lot of variants will be published as pathogenic, and maybe this was before gnomAD even existed, and now we know, well, these variants are actually quite common, so they're probably not causing PCD. So there has to be some data cleaning in terms of the variants. And also because we don't want to include benign and likely benign variants, like I said. So this is kind of a simplified workflow of how we've classified the variants ourselves. So we collect them and then we reclassify them ourselves. This is just to speed up the process. So the first thing that we look at, does it have a high impact in VEP? So VEP is an annotation tool that annotates variants for consequence of the variant on the protein, and high impact is those that I was mentioning that can cause a premature truncation of the protein. And in the case of PCD, we know that those are usually disease causing. So if it is that type of variant, and also it's rare in gnomAD in the general population database, and it hasn't been put on ClinVar as anything other than pathogenic, or if it's not there, then that would make the variant directly to be pathogenic. So this is with high confidence because it needs to meet all of those rules for us to say directly this is pathogenic. But conversely, on the other side of the graph, so if you have a variant that is not high impact in VEP, so maybe a missense variant, for example, it needs to meet a lot more rules for it to be classified as pathogenic. One of them, it will always have to be rare, very, very rare, or absent in NOMAD. And it has to be classified as pathogenic in ClinVar by multiple submitters and no conflicts. So it can't just be one submitter that says it's pathogenic, and it can't be that it has conflicting submissions either. Another rule that I wanted to suggest, but so far I haven't seen, is that instead of being on ClinVar, if it's not, if we do have a very high incidence of the variant on CiliaVar or in our database, then maybe that could push it as well, but that I haven't seen that yet. Maybe one day as we get more and more variants. And if they don't fall into the pathogenic bucket, we do manually classify, but so far most of these have just ended up being VUSs because they don't meet these rules. So another thing that is important to do with the data that we have, besides validating the actual variants, is just validating the data itself in terms of clinical data as well that we get, for example. So we can check for things that we already know. Like Hannah was mentioning, we do know that situs inversus will not be associated with a subset of PCD genes, so we can look at that and see what correlations we can see. So it is good to see that for these three categories, where you can see a 50-50 proportion of patients that have situs inversus versus situs solutus, so this is the laterality defects, we can see more or less a 50% distribution. But we can see that the central pair complex / radial spoke genes, usually these are the ones that we know should not be associated with situs inversus. When I did initial validations, we could see actually there was a small number of patients, so this is unexpected. But once I checked these patients, actually they had benign variants, so we excluded those anyway. So we do have very good data. I also just wanted to address the multiciliogenesis. Usually these genes are not associated with situs inversus, but there is one gene, FOXJ1, which is. So all of these cases with situs inversus have variants in FOXJ1, and that's because FOXJ1 has a role in nodal cilia, which the other multiciliogenesis genes like CCNO and MSitis do not. So our data is good. And maybe this is a little bit, well, this is similar to the last slide, but now this is looking at the ultrastructural defects versus the genotype. So this is another thing, as Hannah was explaining, there is a good correlation in terms of the understanding. But this one isn't perfect, but this does, it is representative of what we do see with EM. It's not a perfect technique. For example, the inner dynean arms are quite difficult to see. But we can see that, you know, for patients that do have outer dynean arm defects, the majority of them in green do have an outer dynean arm gene involved. And for example, here, when you have, for patients that have both missing outer and inner dynean arms, most of them do have a dynean assembly factor gene causing the PCD. So these genes are ones that assemble both the inner and outer dynean arms. So if you have a mutation in that gene, you're missing both arms. So this does correlate with what we know, which does make our data look good. So the database will be online. So we have been, this was a first draft, and this is being maintained and curated by the Health Informatics Center at the University of Dundee. So this will be the interface, and you can search by a gene. You can search by one variant within that gene or a combination of two variants as well. And we did do a first test of the website, and we sent it out to some users and then got some feedback. And now the IT team in Dundee are working through these additional requirements before making it live for everyone to use. But this will be open access for any researchers, clinicians, even patients that are interested to check information on PCD variants. So when you do search for a variant, you get a results page, and it will tell you the amount of individuals within the database have this variant. and it will tell you some of the clinical characteristics that relate to that specific variant. So these are the different things that we include here that you can see. So neonatal respiratory distress, whether they have productive wet cough, also laterality defects and infertility and bronchiectasis, which is the lung function. So this is just to show when you do search for a combination of variants, you will get variant 1. You would get a box here, but because we will only show this for variants where you have more than 5 individuals. So if you have less than 5, it wouldn't show you a box. But you get a box for variant 1, variant 2, and also the combination of variants. And at the bottom of the page, you do get one for the gene overall as well. And I do want to say we have asked for one of the requirements that we want to change is the actual variant nomenclature. It will be the code like the cDNA change with the protein change, which is a lot more user-friendly than this one here. So another requirement that we have asked to be incorporated, which will be, is this variant page. So for each variant, you can click on it and then actually see evidence behind that variant. So you can, this is an example variant, but you can see the classification that we have classified. You can see how many individuals in the database have this variant, how many of them are homozygous, and how many have a second different variant. It will also tell you if the gene is definitively associated with PCD, strongly, moderately, and if you click on this link, it would give you the evidence page from the GSEP that Hannah showed for DNH5. But in this case, it would be the different gene. What type of variant it is. You can also get a link to ClinVar, which is the public variant database that will have more information as well. NOMAD, which is the population database. So it can tell you this variant is present in how many alleles in the general population database. And you do also get in silico predictors. So these are tools that predict the effect of your variant. And lastly, you do get all the publications from which we've included patients in CiliaVar that have this variant as well. We also want to have a gene page. So this is another example of one. Again, this also links to the ClinGen evidence summary for this gene that explains all of the evidence behind the gene association with PCD. It does tell you a bit about the gene. So in this case, our SPH9 is part of the radial spoke head. And you get this kind of information as well, where it does tell you this gene in this case is not associated with situs inversus. Usually you should expect rotating cilia and high-speed video. And in TEM, you would expect a central complex defect, and on IF, you would have absence of the protein as well. And I believe these PMIDs are gene discovery papers as well. So just lastly on CiliaVar, I just wanted to show some of the things that we can do with the data as well. We can try to relate some of the genetic data to the clinical data. And this is an example. So FEV1% predicted is a lung function measure. So this is fixed. It takes into account your things like your gender and your height. So 100% predicted would mean your lungs are very good. So with PCD, you do have a lower FEV1, and we can see the distribution of the genes. And as we already know, CCDC40 and CCDC39, for example, are more severe genes, and we can do this kind of correlation analysis as well. So to summarize, PCD genetics is an increasingly essential part of PCD diagnostics, but is not yet a gold standard test. One of the reasons is because genes keep getting discovered, and as more genes get discovered, we do have more problems with the VUS as more variants come up that we don't understand if they cause disease or not. And also the incidence of PCD is not well understood. It is thought to be an underdiagnosed condition as well. There's many challenges in genetic variant interpretation that we've discussed. And we have the two go-to open access resources that aim to help us resolve the US's and also enrich biological understanding of PCD. So we have the ClinGen that we've discussed, which standardizes gene curations for PCD. And this overall would improve variant classification because we really need to understand whether this gene is a PCD gene before we can even look at the variants. And CiliaVar, the database, which will compile all of the, well, not all, but which will compile PCD-related variants and help us establish these genotype-phenotype correlations as well. I did want to say that we do want to include more data into CiliaVar. So if you have data that you would like to share, that please do reach out to the BEAT-PCD email or to us directly. We are accepting data from centers if you do have ethical approval, but if not, there is a framework that is being built for ethics so that more centers can get involved if you don't have specific ethics to share data as well. And with that, I would like to thank, we would like to thank PCD patients and their families, BEAT-PCD for organizing this talk, Myron and Amelia, the Work Package 2 team, the ClinGen, GCEP, and VCEP teams, the 6 centers that have contributed their data to CiliaVar, the University of Dundee IT team for helping us with the website part of things, and also the Great Ormond Street Children Charity and PCD Support UK for funding my PhD. And thank you all for your attention.
Thank you very much, Motile and Hannah, for your excellent talks, first guiding us through PCD and then genetics and the latest updates. So thank you very much. Excellent. Now we have a lot of questions and also some comments from the audience. We first start with 2 questions, or 3 questions by William Hankey. He says, I'm curious about which of the types of functional evidence you mentioned is considered the best for showing that a certain missense variant in a PCD gene has a deleterious or non-deleterious effect. Do folks in the PCD field have a preference for rescue experiments with zebrafish? Or do they prefer antibody-based studies in patient cells or some sort of multiciliated cell model? Excellent questions.
A very good question. Thank you, Bill. My father and I could both answer. So Bill, I don't know if I acknowledge that Bill's really the lead biocurator for the BCEP and hugely important for the GCEP group. He's really been there right from the word go. So Bill, as you know, we discuss these kinds of things. And actually, the truth is that missense mutations in PCD have had very little actual wet lab experiments done. You can, handfuls of them. So it's really, there aren't very many. And people have done things like get a knockout fish and then overexpress a missense mutation. And that's not really reflecting a recessive disease. So there's lots of problems. You can pick apart anything. It would be great to get more opinions. I suppose I see that Rob even, thanks Rob, has chipped in. I tend to agree that the human model, that would be perfect, showing an actual defect arising in the human, probably patient cells, or maybe to do it in a stem cell where you create the human mutation. Then it didn't have it at all, and you put it in and you can directly see if there's an effect. I don't know, Mafalda, if you want to add anything, but I think it's up for discussion. In a way, being as old as I am, the patient sequencing has really quite taken over. So we don't do much model, well, that's not true, that's an exaggeration, but model organisms get maybe a little bit less emphasis. And so these functional studies it's very difficult to get them funded and so on. But they may be taking even a back step. Yeah. Mafalda, did you want to say anything? I don't know if you have a favorite model.
Not really. I do think that human models are better in general. I think we do tend to prefer those, but I think you said it all, Hannah.
I mean, it's wonderful. The thing about PCD is how striking the variants are. So to get a missense mutation, those have been proven a few times to result in a complete lack of protein. So that's a real, wonderful result. Yeah, we hope for more.
Thank you both very much. And yeah, Rob also agreed with that. A human cell model is the one for him. And we have the next question, Mina.
And the next question is from asking Ceren Ayça. Do you recommend MLPA studies to detect exon deletion and duplications?
Yes, so I have never done an MPLA test, and actually Mamuna has helpfully commented that they may or may not be available. Absolutely. I think, MPLA, I'm correct in saying, doesn't locate your variant for you. It tells you that there's something there. You then have to go and follow up with sequencing. That's right, isn't it? Mamuna, I would love a comment from you. Do you do MLPA?
Yes, I have done once MLPA where I had worked with the company to design the DNA H5 MLPA assay and DNA I1. DNA I1 happens to be a smaller compared to DNA H5, 4 times smaller. So they did for all the exons. For DNA H5, they did they left few exons in the middle, so it can be done only in one assay. And then, if suppose you see the deletion, so single exon deletion for the exon which is not existing would be missed. And MLPA is gene by gene, so it's actually very difficult to design single assay. So right now, the best scenario is that some people are doing bioinformatically with all the gene panels. They tend to do the bioinformatically checking for the CNVs. And I think that would be a better method at this point when we are tackling with so many genes. MLPA will become very difficult and expensive and time consuming, labor intensive.
Thank you.
And I don't think that we have, and I don't think we have MLPA assays for all the genes. Sorry.
Thank you.
And it's true that, I'm so sorry that sequencing doesn't find out, like nothing finds everything, but sequencing, we tend to think it's cheap, but it's not necessarily going to find you your variant.
And MLPA will be much more difficult because, we don't see sequence variants are more common than the CNVs. So.
Excellent. Thank you all for replying, commenting. Now, Rob has a question. How do we correlate cilia or clinical phenotype in CiliaVar when patients have more than one variant in other PCD genes?
I can comment on that.
Yes, Jill.
But that's a good question. I think we haven't really found, I don't think we have in CiliaVar patients that have variants in more than one gene. We have definitely seen it in the literature, but when that's the case, have seen that they just have their monoallelic for one of the genes, so then we wouldn't include that one, and we just include the one that is biallelic. That's kind of been the process to choose in those cases. I've also seen where they do have pathogenic variants in one gene, but then a VUS in another. So it's been added as a note, but the one that we will be correlating is the one with the pathogenic gene.
What if they're both pathogenic, though, Mafalda?
I haven't seen that.
I've got a few of these at the minute. I'll have to talk to you.
Sure.
Thanks.
No, that is very interesting. I haven't seen that. I haven't seen two biallelic pathogenic in two genes. I haven't seen maybe a heterozygous, but not two.
I think the current data. So somebody has said, "Where's the link to CiliaVar? How do I get to use it?" Unfortunately, it's still very much in the developmental stage, and we've got that's. But so BEAT-PCD, thank you so much, has managed to fund the next stage with the IT team, and they have a giant list of things that they're going to improve, as Mafalda touched upon. But it's not really set up in that way. But I just wanted to say that I guess the data that we've got so far is from the literature and is from a selected number of clinical centres. So it's sort of very optimal data. And as we hope more people will submit data, I think we're going to get these rather more challenging cases. But it's not really the case that you would look at. I don't think that the CiliaVar is really set up to look at 2 genes in any way. You'd have to look at these things individually. But I mean, that's quite food for thought, actually.
Thank
Can
you.
I ask one question about CiliaVar? So you said that these are all the patients with biallelic changes only.
No, so we do.
And
Oh, sorry, Mafalda, you should answer this.
no, sorry. No, but we do have dominant genes and X-linked genes as well. I'm not sure if that's what you're asking, but we do have
No.
that.
Okay, that is a good answer. But what I was asking is that when you're talking about the recessive condition, recessive PCD, and when you're talking about biallelic, not every patient you would have been able to do the segregation, right? So you are actually saying 2 variants rather than
Yes.
biallelic.
Yes,
Correct.
absolutely
Correct.
true.
Correct.
Yes, that is
Okay.
very true, Mum.
Yeah.
Yeah.
No, I was wondering, otherwise it would be a smaller data set, and that's fine.
Much
However,
smaller.
yeah.
Yeah,
We
we do
do-
make a note of whether segregation studies was done or not, and we do make a note if we know that they are compound heterozygous or not. But yeah, it's not all of them.
I'm looking forward to it going online
But actually
someday.
seeing it. Me too.
You know?
I think that's a super important that somebody can have 2 variants and they're actually sitting on the same chromosome, and you don't know if you haven't tested mom and dad, and that's such an important, crucial thing. And there's going to be plenty of those in the literature, which is what's been compiled. We've taken out BUSs, but this has not been screened
Okay.
for we haven't taken out ones that don't have segregation. But as my father says, we've made a note.
Thank you so much. It's been a great talk, both Hannah and Mafalda. Yeah.
Thank you.
Thank you.
Thank you. And the next question is from Satening. Thank you for this event. That was too useful. If patient with neonatal respiratory disease, otitis with effusion, recurrent respiratory disease beyond neonatal stage has only 1 mutation, DNH11 gene of unknown significance, he is 11 months old, no opportunity to check for cilia infrastructure. Should we treat as PCD or should we look for other genes responsible for PCD? Thank you.
So I think the question is saying this patient really looks consistent for PCD clinically. There's 1 mutation in a really difficult. So we haven't talked about DNH11 individually, but it's a super difficult one because it's one of those ones where you get normal EM. And I don't know, but maybe there's been no opportunity to look at the motility. Is that what we're saying? So, there would be a classic, a kind of hyper-frequent ciliary beating with DNH11, but not everyone can do high-speed video, and not everyone can do NO. So there's certainly centres that don't have the full range of techniques. So what do? You can certainly, when it is launched, look up that variant in CiliaVar and see if anybody else has reported it. You can look in ClinVar. So there are online resources to try and understand, is that variant that you've got at a very super low frequency that we expect in PCD? So it shouldn't have any kind of frequency in the normal population. And there's lots of normal population databases, but actually those normal population databases are very heavily centric for European, North European. So if this is a patient in a country where there haven't been lots of normal people sequenced yet, we really may not understand whether it's a polymorphism or a proper disease-causing variant. But you can it's an unknown variant. So you can score that using the ACMG guidelines. And if you don't do those, you get in touch with centres that can. With DNH11, there might be a chance to talk to other centres that have things like electron microscopy or tomography is really what you need for DNH11. But so that's a proper challenge. I don't. I think for sure to look at all the known PCD genes in that patient would make sense, and that is simply not diagnostic on its own. So that's the case, and that's a classic case of why we are developing these larger databases to try and give you a bit more of a satisfactory answer. Mafalda, I don't know if you have any other words. It's difficult.
No, I was just going to say, I'm just wondering if it's homozygous or not.
Looks like
But I
one.
was going to say I'm
Only
happy
one.
to look in CiliaVar if we have the variant there. I am happy to do that if anyone wants to send a variant.
Yes, that was my follow-up question. Maybe it would until CiliaVar is launched.
Until it's
Maybe exactly. Maybe you can send it directly to the experts. I think Mafalda gave them email contacts to pass to contact you directly, and then maybe you can then send an email and report back of what if the VUS is on your list. How about that?
Good idea.
Great. Now we have lots of more questions coming in. We have two questions from Mathieu and one from Claire. Mathieu asks, do you think the prevalence of PCD will eventually be closer to CF? And Claire asks, are we close to doing a new prevalence study?
So that's so interesting. And even though it's an amazing paper, there's the Hannah et al. One that I showed, it fully acknowledged itself that it was quite limited. So it only looked at a certain number of PCD genes. It only had a certain amount of data. So I think the conclusion is bigger data is for sure what's needed to know about that. Could we have an equivalent frequency? I mean, CF is really very frequent indeed, but we don't know how many PCD genes we've got. I think seeing Maimuna on the call, being in the world of diagnostics of PCD, it does feel like the number of new PCD genes is tailing off in terms of ones that would have a sort of major effect. But we are literally, during the 4 years of the GSEP, we've seen many new genes being found. So I just don't think we're able to answer that. But I do think every single study on PCD that's genetic has concluded that there's underdiagnosis, under-recognition. In the UK, we're supposed to have 7,000 families, something like that, with PCD based on the incidence, and there's no way that we know all of those. Absolutely not. So if that's repeated around the world, Mafalda, I don't know if you want to say anything. We just don't know, do we? I think it's really quite an interesting idea. We need more sequencing.
Yeah, but I don't know if it will ever reach the CF level. Because it's so genetically heterogeneous. We don't know enough about the penetrance. We don't know about the expressivity. So we are going to miss the patients where people who have 2 hits and they may not have a disease because its penetrance is not known. So I don't know. So I agree with you, Hannah, that we don't know the answer, and I don't know when will we know the answer. But will it be just like CF? CF is actually the higher incidence is because of that one delta F508 mutation, one common mutation. So we don't have something like that in PCD yet.
I should comment because we're collaborating with the MBARC team and with Amelia. I don't know if Amelia is on the call or Erin. So we are currently trying to see whether there are PCD patients kind of hidden in bronchiectasis, and that's a much broader phenotype that affects 1 in 300, 1 in 400 people. It's way more common, and it's more of an adult onset form of bronchiectasis. So I think as Maimuna has said, this underdiagnosis and lack of recognition is tricky because lots of reasons, but the tests are complicated and the interpretations are complicated. So
good question. Very good question.
Very good question.
Thank you. And another question from Tuba. Thanks to speakers for great talks. Some patients have many heterozygous variants on different or the same PCD genes. Is there a compound heterozygosity in PCD like CF?
Compound heterozygous.
Compound
Yes.
heterozygosity.
Yes, absolutely.
Yes.
Yes. So we haven't found digenic inheritance with single gene, but 60 of them at least. And for sure, you can get variants, different variants. Absolutely. It's not all homozygous. I hope we've answered that question correctly.
I think so, yes. One question from me. In ClinGen, there are other work packages as well, for example, focusing on pharmacogenomics. And so is that data then also then correlated or connected with the VU assist, for example, in regards of there is a more advances in personalized medicine, and so is that data connected, or are there plans to do that?
That's a super interesting question. I don't think I know how to answer that. Mafalda's a biocurator. Do we know about that, or I'm looking for other ClinGen people with a bit more know-how?
I'm not sure I completely understood the question, but I don't know of correlation between pharmacogenomics and PCD. I don't know if that's what you're asking.
Well, so
But I
within other working groups within ClinGen who are focusing on that, that hasn't been discussed at our meetings, making direct connections. Bill, do you have a comment?
I think it's a great question. I'm sorry, I'm a little ignorant about it, but I am aware that other ClinGen groups, so in our group, I think we curate a gene for a relationship to PCD. I think in other groups, they may curate for some kind of like downstream benefit from like a drug or something like that. I feel like that might be a future direction for us, but I'm sorry, I don't know enough about the clinical side of.
And you wonder about drugs that would, for example, tackle a class of mutation, be kind of disease agnostic. So this thing will read through all the nonsense mutations, or these things that might be more generally applicable to various diseases. So yeah, that's a great question, Yinting. I have no clue. We will find out.
Excellent. Yes, I think not just for PCD, any new PCD therapies in development, but of course, some especially older people have other morbidities and other medications that they need. I think, a lot of possible interactions need to be studied or developed. All right. So I think these are all the questions. So thank you all very much. Excellent talks from both of you. So that's why the Q&A really kept going on. And we had, I think that was our most attended expert talk, actually. Yes.